Good evening. My name is Leon Chambers, and today we will be discussing Alexander's disease. Alexander's disease is a rare type of leukodystrophy, which is a neurological condition that affects our brains due to a new mutation in the glial fibrillating acidic protein gene, also known as GFAP. This mutation primarily affects the myelin sheath, a protective material made of fatty acids that form a protective casing around nerve cells. It affects white brain matter, nerve fibers which account for 50% of the brain, our central nervous system, which consists of the brain and the spinal cord. Glial fibrillating acting proteins are an intermediate filament responsible for forming networks which provide support and strength to cells. In the brain, GFAP provides protection by forming a myelin sheath around the nerve cells, allowing for proper transmission of nerve signals throughout the body. The central nervous system is comprised of the brain and the spinal cord, which are responsible for regulating functions such as mental functions, physical functions, integrating and responding to sensory information, complex thinking functions, homeostasis, coordinating body function, just to name a few. So white brain matter consists of 50% of our brain, which is primarily comprised of nerve fibers called axons, which are protected by the myelin sheaths. White matter also connects all four lobes of the brain: frontal, temporal, parietal, and occipital. It houses the brain's emotional center. Areas of the brain also depend on white matter. Those areas are the Broca's area, which is responsible for speech production, facial neuron control, and language processing. The Wernicke's area, which is responsible for language. The prefrontal cortex, which is responsible for planning, complex cognitive behavior, personality expression, decision-making, and social behavior. The hippocampus also depends on white brain matter because it is responsible for learning and memory. Alexander's disease is hereditary and can be passed down due to a new genetic mutation in genes, which can occur at any phase of the life cycle. It can occur in the womb, which is called the neonatal phase. It can occur during infancy, which is called the infantile phase. It can occur during the preteen and teenage years, which is called the juvenile phase. And it can occur during adulthood, which is called the adult phase. Alexander's disease has various signs and symptoms depending on the life cycle. The neonatal phase, which is one of the rarest forms of Alexander's disease, usually occurs during the first month of the fetus's life. If the fetus does develop into an infant, the child can be born with severe intellectual and developmental disabilities, have brain fluid buildup known as hydrocephalus, and be prone to seizures. As an infant, which is comprised of 80% of cases, onset usually happens within the first 6 months of life or before the age of 2. Signs and symptoms include hydrocephalus, seizures, poor coordination, bone and joint stiffness, intellectual disabilities, and/or retardation. In the juvenile phase, signs and symptoms are usually displayed between the preteen and early teen years. Symptoms include excess vomiting, difficulty swallowing, poor coordination, loss of motor control, speech problems, inability to cough, muscle spasms, and Alexander's disease can also cause changes in the brainstem. In the adult phase, it is one of the least progressive and aggressive forms of Alexander's disease. The onset usually occurs between the late teen years at any point in adulthood. Young adults will exhibit the same signs and symptoms of those who experience onset of Alexander's disease during the juvenile phase of life. Older adults, due to a weakened immune system, will display signs of ataxia, speech problems, aphasia, and have sleep disturbances. Symptoms are so mild during this time that diagnosis is not rendered until an autopsy is performed and evidence of Rosenthal fibers are noticed by the medical examiner. Alexander's disease is diagnosed 4 ways, either through an MRI, a CT scan, blood testing, or DNA or genetic testing. When examining an MRI to determine if a patient has Alexander's disease, physicians look for the following: extensive cerebral white matter abnormalities with frontal predominance, abnormal signal in the brainstem, specifically in the midbrain and the medulla, swelling or volume loss in the basal ganglia and thalamus. In CT scans, physicians look for decreased attenuation in the frontal white matter. In blood testing, Alexander's is detected by measuring elevated cerebrospinal fluid and elevated GFAP levels. In DNA or genetic testing, lab scientists look for an abnormal copy of the GFAP gene that was inherited from either parent as a result of a new mutation. A mutation could have occurred in the sperm or the ova during the embryonic stage. As far as treatment, currently there is no treatment for Alexander's disease. Symptoms can only be managed with a combination of therapeutic drugs and proper nutrition. Individuals should consult with their physician to discuss possible options. Prevention. Currently, there is no established protocol to prevent this genetic mutation, as both parents could not carry the gene and a baby can still be born with Alexander's disease. This is why a majority of mutations are classified as new, since no one has carried the gene previously. So we do have a few photos here that illustrate a couple of things. So we'll start off with the most popular phrase I have used throughout this entire presentation, which is the myelin sheath. So here we can see this is a neuron. See the nucleus, we see the body, and we see this long line here, which is called the axon and the axon terminal. Now, the axon terminal is the one that sends and receives the signals, and it's like a flow. You have the nucleus, it all works together, goes through here. So it receives a signal here, and it goes through the axon. It goes through the axon here. This is the axon, okay? So you see the myelin sheath? So without this sheath right here, all of the signals that we're trying to send out or receive are disrupted. You see how that protects? And it's the myelin sheath, again, is made of fatty acids. So you have a visual, because I kept saying it, so here you go. Here's a visual representation of the myelin sheath, okay? And also, ta-da! And CT imaging is a CT scan of a brain that is showing signs of Alexander's disease, okay? And where you can see right here, it says CT findings. It is, I have to turn this around to actually read it. Low attenuation in the deep frontal white matter. Basal ganglia may also show low density changes. And enhancement occurs in the basal ganglia and periventricular regions. Hope I said that right. But again, as you can see, this is not what a normal brain should look like. Brain looks like this, then a physician can properly diagnose a person with Alexander's disease. So I hope you have enjoyed the presentation, and any further questions, feel free to shoot an email, and have a good evening.